With the recent announcement that the UK government has approved the use of the Oxford AstraZeneca vaccine, I thought it would be useful just to make a short video to discuss some of the differences between this new vaccine and the previous Pfizer BioNTech one, which was approved back in December. So what's the difference between these COVID-19 vaccines? Just as a very brief recap, genetic material in our bodies is encoded by DNA. That DNA is then transcribed into messenger RNA or mRNA. And the SARS-CoV-2 virus actually is an mRNA virus, so it skips this DNA to mRNA step. So the genetic material encoded by the SARS-CoV-2 virus is already in the mRNA form. This mRNA is then translated into the proteins that make up the SARS-CoV-2 virus. And what scientists have done is they've isolated the mRNA sequence that encodes for the spike protein, which is what you can see circled here. And it's this spike protein that we produce an immune response against. Just to recap, the Pfizer-BioNTech vaccine uses the mRNA approach. So the mRNA is encapsulated or enclosed within a lipid nanobody. So this is just a very small fat particle. And that lipid nanobody allows it to bind with our own cells, releasing the mRNA into those cells, which then go on to produce the spike protein. The Oxford AstraZeneca vaccine uses quite a similar approach, but instead of using a lipid nanobody, it will use what we call an adenovirus vector. Now, this adenovirus vector in this case is actually a virus which can cause a common cold in the chimpanzee. It's been modified so that it cannot cause disease in humans, it cannot replicate in humans, it does not cause COVID-19 in humans, but it is a method through which we can deliver the mRNA to our own cells, and we'll inject the adenovirus vector, which contains the mRNA sequence for the spike protein, and it's this that will deliver the mRNA to our cells. The way it does that is that the adenovirus will bind to our cells and it will enter the cell. Once it's inside the cell, it will release the mRNA, and our own cellular machinery will translate that mRNA into the spike proteins, which are then expressed on the surface of the cell. And it's that we identify as foreign and raise an immune response against. And I've discussed that in a bit more detail in a previous video about the Pfizer BioNTech vaccine, so I'll link that down in the description in case you haven't watched that. As with everything, there are pros and cons of the approach. The big pro of this approach using adenovirus is that it's stable and can be stored in the fridge, unlike the Pfizer BioNTech vaccine, which has to be stored at about -70. With the adenovirus vector, you get a highly specific delivery of the mRNA to our cells, and you also get a high expression of antigen following vaccinations. So that's the spike protein, and therefore we get a robust immune response even from a single dose. The cons are that it takes slightly longer to produce because you also have to grow up the adenovirus in order to deliver the mRNA. And it's possible for some individuals to develop immunity to the viral vector itself. So remember, the viral vector is identified as foreign by our body, and we will raise an immune response against that as well, which can cause some problems if we get a very strong immune response against that viral vector. So it may not be as effective in some individuals. Thank you very much for listening. I hope you found this video useful. If you have, please consider subscribing to the channel, give the video a thumbs up, and if you have any questions, please leave them in the comments section below, and I'll get back to you as soon as I can.